What if the key to our humanity wasn't forged on Earth? Two ancient human populations mysteriously merged around 300,000 years ago after being separated for 1.5 million years. This genetic blend, with one group contributing 80% of modern human DNA and the other 20%, created something unprecedented. A species with cognitive abilities far beyond survival requirements. The sudden appearance of the human-specific FOXP2 gene variant enabled revolutionary language capabilities that transformed human development. This genetic feature is completely absent in Neanderthals and Denisovans. Its remarkable emergence raises a provocative question about the true origin of this vital piece of our humanity. The genetic mystery box. What if the most human part of you, your ability to speak, came from somewhere beyond Earth? When we consider what makes us uniquely human, language stands at the center of our identity. This capacity to communicate complex ideas separates us from all other species on our planet. But the genetic foundation of this ability contains a scientific mystery that challenges conventional understanding of human evolution. At the heart of this mystery lies a gene known as FOXP2. Scientists have identified this gene as a critical controller of human language capabilities and cognitive function. FOXP2 belongs to a family of genes that regulate how other genes are expressed, essentially functioning as a master switch for numerous developmental processes in the brain, with a direct connection to our speech abilities. The human version of FOXP2 differs from the version found in chimpanzees, our closest living relatives, by just 2 amino acid substitutions at positions 303 and 325. These seemingly minor changes had profound effects, altering how neurons connect in regions of the brain responsible for fine motor control and language processing. The resulting neural architecture gave humans unprecedented capabilities for complex communication. When geneticists examine FOXP2 across species, they find something surprising. Most genes show gradual changes across evolutionary timescales, with small modifications accumulating over millions of years. But the human version of FOXP2 appears to have emerged rapidly, with no clear evolutionary predecessors showing intermediate forms between the chimpanzee version and the human version. This anomaly becomes clearer in DNA sequence comparisons between humans and our closest primate relatives. In chimpanzees, the FOXP2 gene produces a protein with 715 amino acids. What's puzzling is that the crucial changes appear to have happened simultaneously, rather than through the gradual step-by-step process that characterizes most evolutionary adaptations. How could such a small genetic change rewire an entire species' ability to communicate? The importance of these specific mutations becomes evident when we examine cases where FOXP2 functions incorrectly. Research on families with inherited speech disorders revealed that mutations in FOXP2 cause severe problems with language production. Affected individuals struggle to make the precise movements needed for clear speech, even though they understand language perfectly well. The most famous case study involves a British family known as the K family. Across 3 generations, about half of the family members suffered from a severe speech disorder. Genetic analysis revealed that all affected individuals shared a single mutation in the FOXP2 gene, providing definitive evidence of its essential role in human communication. Brain imaging studies of affected family members showed abnormal activity in several language-related brain regions, including Broca's area, a region crucial for speech production. These studies confirmed that FOXP2 shapes how the brain processes and produces language at a fundamental level. When scientists place FOXP2 on a timeline of human evolution, they encounter a puzzling gap. The gene appears to have undergone strong positive selection in the human lineage within the last 200,000 years, relatively recently in evolutionary terms. This timing corresponds roughly with the emergence of behavioral modernity in humans, including complex language and symbolic thought. Yet the intermediate forms of FOXP2 that should exist if the gene evolved gradually are mysteriously absent. This apparent sudden appearance represents what scientists call a genetic discontinuity, a break in the expected pattern of gradual evolutionary change. While most traits evolve through small modifications over long periods, FOXP2 seems to have transformed rapidly, like a switch that was suddenly flipped. The question becomes even more intriguing considering that FOXP2 regulates hundreds of other genes involved in brain development and function. Changes to this master genetic switch create cascading effects throughout the developing brain, altering how neurons migrate, form connections, and process information. The human version essentially rewired critical circuits in our brains, enabling the neural architecture necessary for language. Traditional evolutionary theory suggests that complex traits develop gradually through natural selection acting on random mutations. But the human version of FOXP2 appears to have emerged with remarkable speed, transforming primate communication capabilities within a relatively short evolutionary timespan. Some scientists suggest that FOXP2 might have evolved through punctuated equilibrium, where evolutionary changes happen in rapid bursts followed by long periods of stability. Others propose that the gene might have undergone a rare event like a chromosomal rearrangement that produced multiple changes simultaneously. Yet these explanations still leave questions about the precise mechanism and timing. FOXP2 acted as a catalyst for the development of neural circuits specifically optimized for human language, setting our species on a unique evolutionary trajectory. This genetic innovation created the foundation for human culture, enabling the transmission of complex ideas across generations and ultimately leading to technological advancement. The unusual pattern of FOXP2's development invites broader consideration of how such a critical genetic innovation emerged so rapidly. Language doesn't just enable communication. It shapes how we think. The human capacity for abstract thought, planning, and complex problem-solving all depend on language-based cognitive processes. Through language, humans developed cumulative culture, allowing knowledge to build across generations. The FOXP2 story reveals how a seemingly small genetic change can have profound effects on a species' capabilities and evolutionary trajectory. It demonstrates that evolution sometimes takes unexpected paths, producing innovations that transform cognitive abilities. Throughout the human genome, scientists have identified numerous regions showing signs of recent, rapid evolution. Genetic changes strongly favored during the emergence of modern humans. These genetic innovations collectively contributed to making us human, but their origins remain incompletely understood. Two ancient populations, one mysterious. Merger. What caused two separate human populations, isolated for over 1 million years, to suddenly reunite and create modern humans? This question emerges from groundbreaking research at the University of Cambridge that has rewritten our understanding of human origins. Rather than descending from a single ancestral population, modern humans appear to be the product of a mysterious merger between two distinct groups that have been evolving separately for an extraordinarily long time. The research team at Cambridge discovered evidence that human ancestral populations split approximately 1.5 million years ago. This divergence created two separate populations that remained isolated from each other for over 1 million years, developing independently along their own evolutionary paths. Then, around 300,000 years ago, something remarkable happened. These long-separated populations reconnected and merged, creating the genetic foundation of modern humans. This finding represents a significant shift in our understanding of human evolution. Previous models typically described a more straightforward evolutionary tree with populations branching out from common ancestors. This new research reveals a more complex pattern where branches that had grown apart for an immense period eventually rejoined. The scientific mystery deepens when we consider the timing. Why would these populations, after developing in isolation for over 1 million years, suddenly come together around 300,000 years ago? What forces or circumstances drove this merger after such an extended separation? These questions have profound implications for understanding human origins. To uncover this genetic merger, the Cambridge researchers developed a sophisticated computational algorithm called COBRA. Like a genetic detective, COBRA unravels ancient secrets hidden in our DNA by detecting evidence of ancient population splits and mergers through analyzing patterns in modern human genomes. The team tested their algorithm using simulated data before applying it to real human genetic information from the 1000 Genomes Project, a global initiative that sequenced DNA from populations across Africa, Asia, Europe, and the Americas. The algorithm revealed that one population contributed approximately 80% of our genetic makeup, while the second population contributed the remaining 20%. This asymmetric contribution suggests a complex integration process rather than a simple blending of equals. The history of these two populations appears dramatically different. The research revealed that the population contributing 80% of modern human DNA experienced a severe bottleneck immediately following the initial split. This population shrank to a very small size before slowly recovering over approximately 1 million years. This same population also appears to be the ancestral group from which Neanderthals and Denisovans later diverged. This bottleneck event suggests this larger contributing population experienced intense selection pressure or environmental challenges that dramatically reduced their numbers. Such population crashes often accelerate evolutionary changes as genetic traits that enhance survival become more concentrated in the remaining group. The patterns found in the genetic contributions from the minority population revealed that genes from this group were often located away from functional regions of the genome, suggesting potential compatibility issues between the two genetic backgrounds. This pattern indicates that natural selection may have actively removed certain genetic elements, while preserving others. Despite these apparent compatibility challenges, specific genes from the minority population appear to have been strongly selected for retention in the modern human genome. Particularly significant are genes related to brain function and neural processing, suggesting the minority population may have contributed key innovations that shaped human cognitive development. The timing of this merger becomes even more significant when we examine what followed in human evolution. The genetic recombination occurred approximately 300,000 years ago, just before evidence of major cognitive advancements appears in the archaeological record. This temporal relationship raises the possibility that the genetic merger itself may have triggered or enabled the cognitive developments that define modern humans. This coincidence challenges us to consider whether the merger of these two long-separated genetic lineages created novel combinations that enabled unprecedented cognitive capabilities. Could the integration of different genetic backgrounds have produced synergistic effects that neither population possessed independently? The timing strongly suggests this possibility. Traditional natural selection alone struggles to fully explain why specific genes from the minority population were retained despite apparent compatibility issues. If these genes created significant integration challenges, they would need to provide substantial advantages to overcome these barriers and be preserved in the modern human genome. This genetic merger represents a unique event in evolutionary history, rather than the gradual accumulation of small changes typically associated with evolution. The reunion of these long-separated populations potentially introduced substantial genetic innovations in a relatively short time frame. This mechanism could help explain the relatively rapid emergence of modern human cognitive capabilities. The evidence suggests this genetic merger created unprecedented cognitive potential. By combining genetic adaptations that evolved separately for over 1 million years, this reunion may have produced novel neural architectures and cognitive capabilities that neither population possessed independently. What makes this discovery particularly significant is how it relates to the timing of human cognitive evolution. The period following this merger saw the emergence of anatomically modern humans and the gradual development of behavioral modernity, including complex tool use, symbolic art, and eventually, civilization. This research fundamentally changes our understanding of human origins. Rather than evolving from a single ancestral population, modern humans represent the integration of two distinct genetic lineages that developed separately for an extended period before reconnecting. This merger created a novel genetic foundation that enabled unprecedented cognitive development. The question of what drove these populations to merge after such long separation remains unanswered. Geographic changes, climate shifts, or population migrations may have brought these groups into contact. Whatever the specific cause, this reunion set human evolution on a unique trajectory that would ultimately support abstract thinking, complex language, artistic expression, and technological innovation. Beyond survival, the evolutionary surplus. Why do humans possess mental abilities that far exceed what's needed for basic survival? This question strikes at the heart of what makes our species unique and challenges conventional evolutionary theory. In standard evolutionary biology, traits developed to solve specific survival problems. Animals evolve sharper claws to catch prey, thicker fur to survive cold climates, or camouflage to avoid predators. Each adaptation serves a clear purpose. It helps the organism survive long enough to reproduce and pass on its genes. This fundamental principle, natural selection favoring traits that enhance survival, has guided our understanding of evolution since Darwin. Yet human cognitive abilities present a puzzling exception to this rule. Our mental capabilities vastly exceed what should be necessary for survival in our evolutionary environment. Early humans needed to find food, avoid predators, select mates, and raise offspring, tasks that other mammals accomplish without advanced cognition. But our brains evolved capabilities that appear completely disconnected from these basic survival needs. Our minds function like supercomputers designed to run space exploration software when all survival requires is a basic calculator. We perform complex calculations, develop abstract mathematical theories, and conceptualize numbers beyond anything in nature. We create philosophical systems and contemplate metaphysical concepts completely removed from physical reality. Simultaneously, we invest precious energy in artistic expression, cave paintings, musical instruments, and decorative objects, diverting resources from immediate survival tasks. These universal human drives have minimal connection to basic survival yet remain fundamental aspects of our psychology. This cognitive surplus becomes more perplexing when compared to other successful species. Insects have thrived for hundreds of millions of years with simple nervous systems. Reptiles dominated Earth for 165 million years with relatively simple brains. Even mammals like rats and rabbits reproduce successfully without complex cognition. Evolution typically favors efficiency. Organisms develop just enough capability to survive in their ecological niche and no more. The human brain represents an extraordinary biological investment. It consumes approximately 20% of the body's total energy, a significantly higher proportion compared to other species, including our closest primate relatives. This energy demand created a serious evolutionary disadvantage. Every calorie devoted to maintaining our oversized brains was a calorie not available for muscle development, immune function, or reproduction. From an evolutionary perspective, this energy allocation only makes sense if the cognitive abilities it purchased provided proportional survival benefits. But many of our most distinctive mental capabilities appear to offer minimal survival advantage, especially in the resource-scarce environments where humans evolved. This energy-intensive brain would have made early humans more vulnerable to starvation and other environmental challenges. Chimpanzees, with whom we share a common ancestor, have brains that consume a smaller percentage of their total energy. Their lower metabolic cost makes them less vulnerable to food shortages while still allowing sufficient cognition for their survival needs. Natural selection typically favors such efficiency, just enough capability for survival without excessive biological costs. Our language capabilities similarly exceed practical needs. Basic communication aids survival through coordination and knowledge sharing, but humans develop languages with complex grammar, abstract vocabulary, metaphor, and poetry, capabilities that go far beyond simple information exchange. We discuss hypothetical scenarios, distant past events, and potential futures, linguistic abilities that transcend immediate survival concerns. Even our social cognition exceeds practical requirements. While social cooperation enhanced survival for early humans, our ability to maintain complex social networks, track intricate social hierarchies, and develop elaborate cultural systems goes beyond what comparable social mammals require. We understand the mental states of others, anticipate reactions to hypothetical situations, and maintain relationships with individuals we rarely see, cognitive abilities that exceed basic survival needs. Why would natural selection favor such energy-intensive cognitive surplus when simpler brains are more efficient? Traditional Darwinian theory struggles to explain traits that do not directly enhance survival or reproductive success. Several potential explanations exist within conventional evolutionary theory. Sexual selection might have favored intelligence as an indicator of genetic quality, similar to how peacock feathers evolved despite their impracticality. Or perhaps our cognitive surplus emerged as a byproduct of selection for other traits, a phenomenon known as acceptation. Alternatively, group selection might have favored cognitive abilities that benefited entire human communities rather than individuals. These conventional explanations still struggle to account for the magnitude of human cognitive surplus. The gap between necessary survival cognition and actual human capabilities remains surprisingly large. This disconnect suggests either unknown evolutionary pressures we have yet to identify or the possibility of external influence on human development. Whatever its origin, this cognitive surplus enabled humanity's remarkable cultural and technological development. Our ability to imagine possibilities beyond immediate experience, to think abstractly, and to develop cumulative knowledge allowed us to transform our planet in ways no other species has achieved. This cognitive surplus becomes even more suspicious when we examine specific genes found only in humans. While our overall DNA closely resembles that of chimpanzees and other primates, certain genetic elements appear exclusively in humans. These human-specific genes, particularly those involved in brain development, raise a profound question. Why would evolution invest so heavily in mental capabilities that far exceed what's necessary for survival? Nova1, the human-only gene. What explains the existence of a gene variant that appears exclusively in humans but not in any other species, including our closest evolutionary relatives? This question leads us to a remarkable discovery by researchers at Rockefeller University who identified a protein variant that exists only in modern humans. The gene in question is called NOVA1, and its human variant contains a small but significant difference from the version found in every other species on Earth. This human-specific variant is completely absent in Neanderthals and Denisovans despite their extremely close evolutionary relationship to modern humans. These ancient human relatives shared most of our DNA and lived alongside early modern humans. Yet they lack this specific genetic innovation. This absence is scientifically significant because Neanderthals and Denisovans were so closely related to modern humans that interbreeding occurred, with many present-day humans carrying small percentages of Neanderthal or Denisovan DNA. The human lineage diverged from these archaic humans roughly 500,000 to 750,000 years ago for Neanderthals. For a genetic variant to exist exclusively in modern humans suggests a recent and significant evolutionary development. The discovery emerged from a comprehensive genomic analysis comparing 8 human genomes with 3 high-coverage Neanderthal genomes and 1 high-coverage Denisovan genome. This comparison confirmed that the NOVA1 variant found in modern humans was absent in all examined archaic human samples, strengthening the conclusion that this variant is truly unique to modern humans. NOVA1 functions as an RNA-binding protein with a crucial role in how genes are expressed in the brain. It helps regulate alternative splicing, a process that allows a single gene to produce multiple proteins. This function makes NOVA1 particularly important for neural development and neuromuscular control, determining which proteins are produced in different brain regions. A study published in Nature Communications demonstrated NOVA1's significance through an innovative experiment. Researchers used CRISPR gene editing to replace the standard NOVA1 protein in mice with a human-specific variant. The mice with the human NOVA1 variant showed altered vocalization patterns. Both mouse pups and adult males produced different ultrasonic squeaks compared to normal mice. The human variant changed the letters of the squeaks, essentially altering the basic sound units used for communication. Notably, the mice with the human variant communicated differently with female mice, suggesting changes in social communication patterns. These altered vocalizations provide compelling evidence that NOVA the first of may be directly involved in communication development. The researchers discovered that the human NOVA1 variant affected RNA binding specifically related to vocalization. The binding sites most substantially affected were located at genes coding for RNAs associated with vocalization, explaining how this single genetic change could influence communication abilities. NOVA1's timing remains uncertain, but its absence in Neanderthals and Denisovans indicates it emerged within the last 500,000 years, coinciding with the period when modern humans were developing increasingly sophisticated cognitive abilities, including complex language. NOVA1 becomes even more fascinating when compared with FOXP2, often described as a potential language gene. While FOXP2 has received more scientific attention, NOVA1 represents another critical piece in the language puzzle. Like finding a key piece missing from all other species' genetic puzzles, NOVA1 appears exclusively in the human lineage that developed complex speech. FOXP2 differs between humans and chimpanzees by just 2 amino acids, yet these small changes significantly affect language development. Similarly, NOVA1's human variant creates substantial effects through minimal changes. Together, these genes suggest human language capabilities emerged through multiple genetic innovations rather than a single language gene. Modern humans likely developed their unique communication abilities through the cumulative effects of several genetic changes, including both FOXP2 and NOVA1 variants. Why would such a significant gene variant appear only in humans? Under conventional evolutionary understanding, genetic innovations typically develop gradually across related species, with closely related species showing similar adaptations. NOVA1's exclusive presence in modern humans represents a discontinuity in this pattern. Several possibilities might explain this anomaly. The NOVA1 variant may have arisen through mutation after humans diverged from Neanderthals and Denisovans, then was strongly selected for due to communication advantages. Alternatively, the variant might have existed in the common ancestor population but was lost in Neanderthal and Denisovan lineages while being retained in modern humans. However, the timing and specificity of this genetic innovation raises questions about conventional evolutionary mechanisms. The variant appears precisely in the lineage that develops sophisticated language, affecting genes specifically related to vocalization. This specificity suggests targeted adaptation rather than random genetic drift. By influencing RNA binding and alternative splicing in the brain, NOVA1 shapes neural architecture that enables complex language, affecting how neurons develop and connect, processes fundamental to language processing neural circuits. NOVA1 represents a genetic anomaly that challenges conventional evolutionary explanation. Its exclusive presence in modern humans, absence in closely related species, and effects on vocalization-related genes create an evolutionary puzzle. The gene emerged in the lineage that developed complex language, affecting the exact biological processes needed for sophisticated communication. These genetic anomalies become even more puzzling when we consider how they've shaped the language lateralization enigma. How did humans develop a brain structure for language that's fundamentally different from every other species on Earth? While many animals communicate through various means, birds sing, whales create complex vocalizations, and primates use gestures and calls, only humans possess a neural architecture specifically optimized for language processing. This structural difference represents one of the most distinctive features of the human brain and creates a profound evolutionary mystery. The human brain exhibits a phenomenon known as language lateralization. This term refers to how language processing is concentrated primarily in the left hemisphere of our brains. Imagine the left hemisphere as a specialized control center with dedicated stations for different language functions, each one optimized for specific tasks like speech production or comprehension. While both hemispheres participate in language, the left hemisphere contains specialized regions like Broca's area and Wernicke's area that play critical roles in speech production and language comprehension, respectively. This asymmetric organization creates a dedicated neural network optimized for language processing. What makes this lateralization particularly interesting is its uniquely human nature. When comparing human brains with those of other primates, including chimpanzees and bonobos, researchers have failed to find equivalent language-specialized regions. Other primates show some brain lateralization for certain functions but lack the specific pattern of left hemisphere language specialization that characterizes human brains. Functional neuroimaging reveals this difference clearly. When humans process language, specific regions in the left hemisphere show increased activity, forming a specialized network that includes Broca's area in the frontal lobe, Wernicke's area in the temporal lobe, and the arcuate fasciculus, a bundle of nerve fibers connecting these regions. This neural circuit forms the foundation for human language capabilities, allowing us to process grammar, syntax, and meaning with remarkable efficiency. What if our brains were wired completely differently? The answer becomes apparent when examining the structural differences in language-related areas between species. In humans, Broca's area shows distinctive patterns of neuron types and arrangements that differ from corresponding brain regions in other primates. Similarly, the arcuate fasciculus is significantly enlarged and more complex in humans, creating enhanced connectivity that supports complex language processing. The FOXP2 gene, previously discussed as critical for language development, appears associated with this lateralization process. FOXP2 influences the development of asymmetric neural circuits in language-related brain regions during early development. The human variant promotes the specific pattern of left hemisphere specialization that enables complex language processing. This connection between FOXP2 and brain lateralization becomes particularly evident in cases where the gene functions abnormally. Individuals with FOXP2 mutations show altered activation patterns in language-related brain regions and often have reduced lateralization. Their brains process language using more distributed neural networks rather than the specialized left hemisphere circuit typical in most humans, resulting in significant speech and language difficulties. The lateralized language network creates a hierarchical processing system that handles different aspects of language efficiently. Lower-level processing in the superior temporal gyrus analyzes phonemes, basic sound units, while higher-level processing in Broca's area manages grammar and syntax. This architecture allows humans to process language with unmatched speed and accuracy. Human language areas contain specific types of neurons, including von Economo neurons and a higher density of dendritic spines, that enhance connectivity and information processing capabilities. These cellular adaptations create neural circuits capable of processing complex syntactic structures that characterize human language. This specialized architecture raises a profound question. Why did human brains develop this unique organization when other communication systems in nature don't require it? Communication exists across animal species, yet none developed a specialized, lateralized neural architecture comparable to the human language network. The question becomes more puzzling considering that lateralization increases processing efficiency but requires significant evolutionary restructuring. Brain lateralization offers several advantages, reducing duplication of neural functions, allowing parallel processing of different information types, and minimizing interference between potentially competing processes. These benefits explain why some degree of lateralization appears across many species. However, the extreme specialization in human language lateralization represents a significant evolutionary investment. It required substantial restructuring of the primate brain, rededicating neural resources and establishing new connection patterns. From an evolutionary perspective, such restructuring would only occur under strong selection pressure. Yet the selective advantages of early language capabilities remain unclear. This specialized architecture comes with a potential vulnerability. Highly specialized neural systems can be more susceptible to damage than distributed networks. A stroke affecting Broca's area or Wernicke's area can severely impair language abilities, while damage to corresponding regions in the right hemisphere typically has minimal effects on language function. Despite this vulnerability, human brain lateralization for language persists as a universal feature across populations. While the degree varies somewhat between individuals, the pattern of left hemisphere language specialization appears in approximately 95% of right-handed people and about 70% of left-handed individuals. This consistency suggests the advantages substantially outweighed any disadvantages during human evolution. Human brain lateralization represents an architectural leap with no clear evolutionary precedent. While brain asymmetries exist in other species, the specific pattern and degree of language lateralization in humans represents a novel neural architecture that emerged relatively recently in evolutionary history. This specialized organization developed in concert with genetic innovations like human variants of FOXP2 and NOVA1, creating the neural foundation for human language capabilities. The cognitive explosion timeline. What triggered the sudden explosion of human cognitive abilities after millions of years of relatively slow evolution? When we examine the timeline of human development, we find a pattern that challenges conventional evolutionary understanding, a pattern of gradual change suddenly interrupted by an acceleration that transformed our species. For millions of years, hominin cognitive development proceeded at a measured pace. Our ancestors like Homo habilis and Homo erectus showed incremental improvements in tool making, with basic stone tools remaining largely unchanged for hundreds of thousands of years. Brain size increased gradually, from approximately 600 cubic centimeters in early Homo species to around 1,000 cubic centimeters in archaic Homo sapiens. These changes occurred over vast timescales consistent with traditional evolutionary processes. The archaeological record from this extended period shows consistent behavioral patterns. Early humans used simple stone tools, controlled fire, and lived in small social groups. Their technology evolved slowly, with innovations like the Acheulean hand axe remaining essentially unchanged for over 1 million years. This stability indicates cognitive capabilities that improved incrementally rather than through sudden leaps. This gradual development pattern changed dramatically around 70,000 to 50,000 years ago. During this relatively brief window, essentially a blink in evolutionary terms, human cognitive capabilities accelerated at an unprecedented rate. This period, often called the Great Leap Forward, or the Upper Paleolithic Revolution, saw a burst of creative brilliance that transformed human behavior in ways difficult to explain through standard evolutionary timescales. The archaeological evidence reveals a sudden flowering of complex behaviors indicating fundamentally new cognitive abilities. Sites from this period contain an explosion of symbolic and artistic expression alongside remarkable technological innovations. Cave paintings appeared, featuring sophisticated depictions with understanding of perspective and anatomy. Humans began creating composite tools combining multiple materials and specialized hunting implements like spear throwers. Most significantly, they produced objects with purely symbolic functions, beads, pendants, and figurines like the famous Venus statues found across Europe. This dramatic shift in material culture represents the emergence of minds capable of abstract thought, symbolic representation, and creative expression. This cognitive revolution coincides with human migration out of Africa. Evidence indicates that modern humans began spreading from Africa to Eurasia around the same period when these new cognitive abilities appear in the archaeological record. These expanding populations brought their new capabilities with them, establishing communities across continents. The timing raises intriguing questions. Did new cognitive abilities enable this expansion, Or did migration's challenges drive cognitive development? The correlation suggests a relationship, though the causality direction remains unclear. What seems evident is that something fundamental changed in human cognitive capabilities during this critical period. Research on the Great Leap Forward theory attempts to explain this sudden acceleration, proposed by paleontologist Richard Leakey and others. This theory suggests a significant shift occurred in human cognitive abilities that enabled complex language, abstract thinking, and cultural innovation. The theory explores potential triggers for this cognitive explosion. Some researchers suggest genetic mutations affecting brain function emerged and spread rapidly due to selective advantages. Others propose existing cognitive capabilities reached a critical threshold, enabling new forms of thinking. Additional hypotheses point to environmental pressures or social changes driving rapid adaptation. Why did this acceleration happen after hundreds of thousands of years of slower development? This timing presents a significant puzzle for evolutionary biology. If anatomically modern humans existed for roughly 200,000 years before this cognitive explosion, what triggered the relatively sudden change? Why didn't these advanced abilities emerge earlier? The question becomes more perplexing when comparing this cognitive explosion to normal evolutionary timescales. Major evolutionary innovations typically require many generations to become established. Complex adaptations generally develop incrementally over extended periods, with natural selection gradually refining advantageous traits. For context, the evolution of the eye occurred over hundreds of millions of years through incremental improvements. This gradual process aligns with traditional Darwinian evolution. In contrast, the human cognitive explosion occurred within perhaps 20,000 years, an extraordinarily brief period evolutionarily. This accelerated development challenges conventional understanding and suggests either unknown mechanisms or external influences. Traditional explanations propose this acceleration might result from cultural rather than biological evolution. Once basic language developed, knowledge could accumulate across generations through cultural transmission, creating a positive feedback loop where improved communication enabled faster innovation. However, this explanation still requires initial biological changes enabling complex language and abstract thinking. The question remains, what biological innovations created the foundation for this cultural acceleration, and why did they emerge when they did? This cognitive acceleration represents an evolutionary anomaly that traditional Darwinian theory struggles to fully explain. While natural selection and genetic mutation played roles in human cognitive evolution, the timing and pace of the Great Leap Forward suggest additional factors may have influenced human development during this critical period. The evolutionary leaps in human cognition present a genuine scientific puzzle. Human cognitive development experienced a remarkable acceleration that transformed our species from clever primates into beings capable of art, complex language, technological innovation, and eventually, civilization. When FOXP2 goes wrong, the language connection. What happens when the alien gene in our DNA malfunctions? To understand the profound impact of FOXP2 on human language, we need to examine cases where this critical gene doesn't work properly. The most illuminating example comes from a British family known in scientific literature as the K family, whose unique genetic condition has revolutionized our understanding of language's genetic foundations. The Ke family presents a remarkable case study spanning 3 generations. Nearly half the family members across these generations suffer from a condition called developmental verbal dyspraxia, a disorder that severely impairs speech production and language processing. Affected family members struggle with basic grammar, have limited vocabulary, stutter frequently, and experience significant pronunciation difficulties. These speech problems persist despite normal intelligence in other areas, suggesting a highly specific language impairment rather than general cognitive disability. What makes this family scientifically significant is that their condition stems from a single genetic mutation. After extensive genetic analysis, researchers discovered the culprit, a specific mutation in the FOXP2 gene located in a chromosomal region designated as SPCH1. This region contains approximately 70 genes, most related to speech development and cognition, underscoring FOXP2's placement within a cluster of genes critical for human communication. The mutation itself is remarkably precise, a single nucleotide change from G to A in the genetic code, resulting in an amino acid substitution from arginine to histidine at position 553 of the FOXP2 protein. This tiny alteration creates cascading effects throughout the brain's language systems. Brain imaging of affected K family members reveals structural and functional abnormalities in language-related brain regions, particularly in the basal ganglia, crucial for coordinating fine motor movements required for speech, and Broca's area, essential for speech production. The selective nature of these abnormalities is striking. Affected family members can perform complex physical tasks requiring fine motor coordination, yet cannot consistently produce the precise motor sequences needed for clear speech. The mutation disrupts motor planning, the brain's ability to coordinate the rapid, precise movements of lips, tongue, jaw, and vocal cords. Speaking requires extraordinary precision, with dozens of muscles working in millisecond-level coordination. When FOXP2 functions incorrectly, this intricate coordination breaks down, making it difficult to produce consistent words and syllables. FOXP2 plays a critical role in neurite outgrowth, the process by which developing neurons extend projections to form connections. The human variant enhances this process specifically in brain regions associated with language. When mutated, as in the KE family, these neural connections develop abnormally, compromising language processing and speech production. This selective impact raises profound questions about FOXP2's evolutionary significance. A single gene mutation specifically impairs language while leaving other cognitive functions intact, suggesting FOXP2 played a specialized role in human evolution, possibly emerging as a key genetic innovation that enabled our unique language capabilities. At the microscopic level, FOXP2 influences the development of medium spiny neurons in the basal ganglia, cells particularly important for procedural learning, the type of learning involved in acquiring automatic motor skills like speech. With mutated FOXP2, these neurons develop abnormally, compromising the brain's ability to automate the complex motor sequences required for fluent speech. While FOXP2 mutations create specific language impairments, this gene is not solely responsible for all language ability. Language processing involves complex neurological networks influenced by multiple genes. FOXP2 functions more as a master regulatory switch, influencing the expression of numerous other genes involved in brain development. This regulatory role explains how a single mutation can have such wide-ranging effects on language capabilities. The KE family case demonstrates how a single gene mutation can specifically target language, one of humanity's most distinctive traits, while leaving other aspects of cognition relatively intact. This pattern suggests FOXP2 evolved specifically to enable the neural architecture required for human language, functioning as a genetic foundation for our species' unique communication abilities. The significance increases when we consider that language represents one of the most distinctively human traits. No other species possesses true language with grammar, syntax, and unlimited generative capacity. The fact that a single gene can so profoundly affect this uniquely human capability raises important questions about FOXP2's origins in evolutionary history. The mutation affects more than speech production. Affected individuals also struggle with language comprehension and grammar processing, suggesting FOXP2 influences neural circuitry for both producing and understanding language. This comprehensive impact further highlights the gene's central role in human linguistic capabilities. When functioning properly, FOXP2 helps orchestrate the development of neural circuits specialized for language processing and speech production. When mutated, these circuits develop abnormally, compromising one of humanity's most distinctive capabilities. The KE family's condition provides compelling evidence that FOXP2 represents a critical genetic foundation for human language. The selective nature of their impairment suggests this gene played a specialized role in human evolution, possibly emerging as a key genetic innovation that enabled our species' unique communication abilities. How can a single gene have such profound effects on a complex trait like language? This pattern suggests FOXP2 sits at a critical junction in the genetic networks controlling brain development, functioning as a regulatory hub that influences numerous other genes involved in neural development. The KE family reveals FOXP2 as a genetic keystone of human language, a master regulatory gene that helped enable our species' unique communication capabilities. When this gene malfunctions, it compromises the neural foundations of language, demonstrating its essential role in what makes us distinctively human. The purifying selection puzzle. Why did natural selection actively remove certain genetic elements after the mysterious merger of ancient human populations? This question leads us to examine one of the most fascinating aspects of human evolution: how our genome was shaped after two long-separated populations reconnected to create modern humans. In evolutionary biology, purifying selection refers to a process where natural selection actively removes harmful genetic mutations from a population over time. This mechanism serves as a genetic quality control system, eliminating variations that could negatively impact survival or reproduction. Purifying selection maintains the integrity of the genome by preserving beneficial genetic sequences while removing detrimental ones. Recent research published in Nature Genetics revealed something remarkable about the merger of two ancient human populations that occurred approximately 300,000 years ago. After these populations reconnected, purifying selection acted with unusual strength specifically on genes from the minority ancient population, the group that contributed roughly 20% to the modern human genome. This selective pressure suggests these genetic elements face challenges integrating with the majority genetic background. The pattern of genetic integration that emerged shows genes from the minority population were often located away from functional regions of the genome. Functional regions are areas that directly code for proteins or regulate gene expression. The positioning of minority population genes in less functional genomic regions indicates these genetic elements may have been less compatible with the majority genetic background. This positioning pattern reveals important information about the ancient genetic merger. The evidence points to a complex process where certain genetic elements from the minority population faced compatibility challenges when combined with genes from the majority group. This selective positioning away from functional regions likely served as a buffer, reducing potential conflicts between genetic elements that had evolved separately for over 1 million years. These compatibility issues make evolutionary sense considering the extended separation of these populations. With approximately 1.5 million years of independent evolution, these groups developed distinct genetic adaptations suited to their specific environments. When these differently adapted genomes combined, conflicts arose, creating selection pressure to resolve genetic incompatibilities. Scientific models examining how purifying selection shaped modern human genomes suggest this process was both powerful and targeted. Analysis of genetic data from diverse modern human populations shows consistent patterns of selection against certain minority population genetic elements. This consistency across geographically dispersed human groups indicates the selection occurred early in the history of the merged population, before humans dispersed globally from Africa. The strongest purifying selection targeted genes related to neural development, immune function, and reproduction, systems where precise genetic coordination is essential. These biological systems demand high levels of genetic integration, with multiple genes working together in complex networks. When genes from two long-separated populations needed to work together in these delicate systems, selection eliminated combinations that functioned poorly. Despite apparent incompatibility with the majority genetic background, certain genetic elements from the minority population were preserved. From an evolutionary perspective, these genetic variants were likely retained because they provided significant advantages that outweighed any disadvantages. These preserved genes offered substantial benefits to the merged population. The most likely explanation is that these preserved genes provided crucial advantages not present in the majority population. Rather than being eliminated, these beneficial genes were retained despite integration challenges. This selective retention created a unique genetic blend combining advantageous elements from both ancestral populations. Research indicates that many preserved minority population genes relate to brain function and neural processing. This pattern suggests the minority population may have contributed genetic innovations related to cognitive function that the majority population lacked. When combined with the genetic background of the majority population, these cognitive innovations may have created novel neural capabilities. The process resembles technological integration more than typical evolutionary change. When two distinct technologies combine, the initial integration often creates conflicts requiring modifications to both systems. Similarly, natural selection removed problematic combinations while preserving beneficial ones, acting like an editor refining a cosmic manuscript by removing misprints while preserving the valuable text. This purifying selection process represents a sudden genetic restructuring driven by the combination of two distinct genetic lineages. The resulting selective pressures shaped the modern human genome in ways that potentially accelerated our cognitive evolution. The timing of this genetic integration corresponds with the period preceding major cognitive developments in human evolution. This temporal relationship suggests that the genetic merger itself, and the subsequent purifying selection, may have contributed to the cognitive capabilities that define modern humans, By combining genetic innovations that evolved separately in two populations, the integration created novel genetic combinations that neither original population possessed. The subsequent purifying selection refined this genetic blend, eliminating problematic combinations while preserving beneficial ones. The study published in Nature Genetics challenges long-standing beliefs about the unpredictability of evolution. The findings suggest that the evolutionary trajectory of a genome may be influenced by its evolutionary history rather than determined solely by historical accidents. This perspective provides a new framework for understanding human evolution after the population merger. How has natural selection shaped these potentially non-terrestrial genetic elements? While the research doesn't explicitly support extraterrestrial origins, it reveals an unusual evolutionary process that shaped the modern human genome. Purifying selection acted as a refining mechanism, creating a genome optimized for unprecedented cognitive potential. This selection process produced a genetic foundation capable of supporting advanced language, abstract thought, and technological innovation, capabilities that distinguish humans from all other Earth species. The evidence clearly shows these genetic elements underwent intensive selective refinement after the population merger. These genetic anomalies force us to consider alternative explanations for human origins. The unusual patterns of genetic integration, selective retention of specific genes, and the timing relationship with cognitive advancement suggest the extraterrestrial hypothesis. Could the unexplained aspects of human genetics have an origin beyond Earth? Throughout our exploration, we've encountered multiple genetic and evolutionary anomalies that challenge conventional scientific understanding. The sudden appearance of the human FOXP2 variant with its critical role in language development. The mysterious merger of two ancient human populations after 1.5 million years of separation. The cognitive abilities that far exceed survival needs. The human-specific Nova-1 gene absent in our closest evolutionary relatives. The unique brain lateralization pattern found only in humans. The unexplained acceleration of cognitive development after millions of years of gradual change. The selective effect of FOXP2 mutations on language capabilities. The unusual pattern of purifying selection that shaped the modern human genome. These anomalies collectively create a scientific puzzle. Each might be explained individually through conventional evolutionary mechanisms, but their combination, particularly their timing and specificity, raises questions about whether traditional evolutionary theory fully accounts for human origins. What if the missing pieces of this puzzle lie beyond our planet? This pattern of evidence has led some researchers to consider a radical alternative explanation, the extraterrestrial hypothesis. This proposes that aspects of human genetic development might have been influenced by non-Earth origins. This doesn't mean aliens in the popular sense, but suggests our genetic material might not be entirely terrestrial in origin. While speculative, this hypothesis addresses puzzling aspects of human evolution that conventional theories struggle to explain. How might extraterrestrial genetic material have reached Earth? Scientists have proposed several mechanisms, with panspermia being the most established. Think of panspermia as cosmic mail delivering the blueprints of life across vast distances. This theory suggests that life's building blocks could travel between planets via meteorites, comets, or interstellar dust. These space travelers might carry genetic components that could influence evolutionary development on their destination worlds. Recent discoveries support this possibility. Researchers have found complex organic molecules, including amino acids, in meteorites that have landed on Earth. Laboratory experiments have shown that some microorganisms can survive the harsh conditions of space, especially when protected inside rocks or ice. These findings suggest biological material could potentially journey between planets or even star systems. A more specific theory is directed panspermia, the intentional seeding of genetic material by an advanced civilization. First proposed by molecular biologists Francis Crick and Leslie Orgel, this hypothesis suggests an advanced extraterrestrial civilization might have deliberately sent biological material to Earth. This could explain the sudden appearance of critical genes and cognitive capabilities in the human lineage. If specific genetic elements were introduced into early human ancestors, it might account for the apparent discontinuities in our evolutionary record. What genetic evidence points to potential non-Earth origins? The strongest indicators come from genetic elements that appear suddenly in the human lineage without clear evolutionary precedents. The human variants of FOXP2 and NOVA1, crucial for language and cognitive function, emerged with remarkable specificity and timing. The unusual genetic integration following the Merger of ancient human populations suggests a process unlike typical evolutionary development. The accelerated cognitive development occurred at a pace difficult to explain through conventional evolutionary timescales. Scientific opinion varies widely on these possibilities. Proponents argue the extraterrestrial hypothesis provides a coherent explanation for multiple evolutionary anomalies. They highlight the genetic elements that appear without clear predecessors yet play crucial roles in distinctively human capabilities, and the unusual timing of our cognitive development, stable for hundreds of thousands of years before rapidly accelerating within a relatively short time frame. The dark forest hypothesis offers insight into why we haven't detected extraterrestrial civilizations despite decades of searching. Like prey animals hiding from predators in a dark forest, advanced civilizations might conceal their presence. Fearing contact could lead to existential threats. This perspective might explain why extraterrestrial civilizations remain undetected. They may actively avoid detection for self-preservation. Skeptics correctly point out that extraordinary claims require extraordinary evidence, and direct proof of extraterrestrial influence remains absent. They argue that conventional mechanisms like natural selection, genetic drift, and mutation can potentially explain human genetic development without external influences. Many maintain that gaps in our understanding result from incomplete evidence rather than extraterrestrial intervention. Most mainstream scientists emphasize that while aspects of human evolution remain unexplained, these mysteries don't necessarily require extraterrestrial explanations. The history of science shows that many once mysterious phenomena eventually received conventional explanations as research advanced. A balanced perspective acknowledges that while the extraterrestrial hypothesis remains speculative, it addresses questions that conventional theories haven't fully resolved. Research into human genetic origins continues, with new techniques providing increasingly detailed insights into our evolutionary history. Whether our genetic uniqueness stems entirely from earthbound processes or includes some extraterrestrial influence, the question transforms how we conceive of humanity's origins. This possibility raises profound questions about human identity and our place in the universe. The implications extend beyond scientific curiosity to touch on fundamental aspects of how we understand ourselves and our relationship to the cosmos. Redefining humanity, the philosophical implications. How would partial extraterrestrial origins change our understanding of what it means to be human? This question moves beyond scientific curiosity into the realm of philosophy, challenging our most fundamental assumptions about identity and existence. If evidence confirmed that crucial elements of our genetic makeup originated beyond Earth, we would face profound questions about our nature and place in the cosmos. Throughout human history, our identity has been tied to origin stories. Whether through religious creation narratives, scientific evolutionary accounts, or cultural mythologies, humans have defined themselves through stories of where we came from. Consider how many of us grew up with bedtime stories about our ancestors or creation myths that explained our existence. These narratives provide a framework for understanding our relationship to other species, our moral responsibilities, and our purpose. They answer the fundamental question: what makes us human? The possibility of extraterrestrial genetic influence disrupts these established narratives. If key genetic elements that enable our most distinctive traits, language, abstract thinking, creative expression, originated beyond Earth, we would need to reconceptualize human uniqueness. Our self-perception as a purely terrestrial species evolving through conventional biological processes would require significant revision. This disruption would affect scientific understanding and cultural and religious frameworks that have shaped human identity for millennia. This reconsideration extends to philosophical traditions across cultures. Western philosophical thought has emphasized human exceptionalism through Aristotle's definition of humans as rational animals and Kantian notions of moral autonomy. Meanwhile, Buddhist philosophy emphasizes the continuity between humans and other sentient beings, focusing on consciousness as a shared quality. These contrasting viewpoints raise a compelling question. If our cognitive uniqueness came partly from the stars, would that make us more special or less special in the cosmic order? Indigenous philosophies often describe humans as emerging from the earth itself, creating a direct kinship with the land and other species. When a Native American elder speaks of humans being formed from clay or emerging from underground, these aren't mere metaphors but foundational beliefs about our connection to this planet. The possibility of extraterrestrial genetic influence would challenge these Earth-centered narratives while potentially reinforcing concepts of cosmic interconnectedness. Religious perspectives on human exceptionalism often emphasize human special relationship with divine forces. In Abrahamic traditions, humans are described as created in God's image. Would the discovery of alien genetic influence in our DNA be seen as evidence against divine creation? Or might believers interpret it as simply revealing another aspect of God's method? What happens to human exceptionalism if our most distinctive traits originate partly from beyond Earth? This question challenges us to reconsider what makes humans unique. If our most celebrated capabilities stem partially from extraterrestrial genetic elements, we might need to rethink our relationship with other Earth species. Rather than seeing ourselves as the pinnacle of terrestrial evolution, we might recognize our position as more complex, simultaneously connected to Earth's evolutionary processes and influenced by factors beyond our planet. The implications extend to our concept of cosmic significance. The Copernican revolution displaced Earth from the center of the cosmos, while evolutionary theory connected humans to other species through common ancestry. Extraterrestrial genetic influence would represent another shift in this ongoing reassessment, suggesting that humanity might be part of a larger cosmic story extending beyond Earth. This possibility raises questions about human purpose. Were our cognitive capabilities random evolutionary developments, or might they reflect some form of cosmic intent? This fundamentally challenges how we understand human development and potential. Our ethical frameworks might also require reconsideration. If human cognitive capabilities partially originated beyond Earth, we might need to expand our ethical considerations beyond terrestrial relationships. The possibility raises questions about potential responsibilities to extraterrestrial life forms that might share genetic connections with us. It also challenges us to reconsider our ethical responsibilities to Earth if we aren't solely products of terrestrial evolution. Despite these profound implications, an important philosophical insight emerges. Regardless of origins, our genetic makeup defines our capabilities but not our values or choices. Even if certain cognitive abilities resulted from extraterrestrial genetic elements, how we use these capabilities remains a human choice. Our capacity for language enables both poetry and propaganda. The origin of these capabilities doesn't determine how we employ them. This distinction between capabilities and choices highlights an essential aspect of human identity that transcends questions of genetic origin. Our humanity ultimately resides not in where our genes originated but in what we choose to do with the capabilities they provide. If our DNA contains alien origins, it doesn't diminish our humanity but rather expands our understanding of it. It places us within a potentially larger cosmic context while maintaining our connection to Earth's evolutionary processes. It challenges us to develop more inclusive concepts of identity that embrace both our terrestrial relationships and possible cosmic connections. The possibility of extraterrestrial genetic influence invites us to expand our perspective beyond earthbound thinking to consider our place in a potentially connected cosmic community. It challenges us to develop frameworks of human identity that accommodate new discoveries while preserving the essential insights of our philosophical, religious, and cultural traditions. Most fundamentally, it reminds us that the question, what makes us human, remains open to ongoing exploration and discovery. The mystery of human origins remains open. We've explored genetic anomalies like FOXP2 and NOVA1, ancient population mergers, our cognitive surplus, and unique brain lateralization, all challenging conventional evolutionary theory. These mysteries persist despite intensive scientific investigation. The answer to our genetic uniqueness will fundamentally transform humanity's self-understanding. Each new discovery opens pathways to further questions while gradually illuminating our past. Are we truly alone in our origin? The truth awaits discovery, perhaps already encoded within our DNA, a cosmic signature hidden in the elegant double helix that defines us. The story of humanity may be far more